methylation, or thought to be modulated by DNA methylation in vivo. All right, so I'll stop on transcription there. Basically, if I've hopefully convinced you of anything, transcription is complicated, and we're more and more able to artificially influence transcription either by using scaffolds or by using enzymes. And I sort of think of us as doing this now, where we're using single domains to sort of blindly or bluntly control transcription, and, but we're assembling toolboxes, I think, where we can combine these in very sophisticated ways to control transcription or epigenetic marks in a way that is much more similar to the way these things are wired during development. And I think this will be really useful for studying disease and thinking about therapies, and in general moving forward with moving away from the nucleus being this scary black box that nobody wants to work on because it's too hard to control any of the processes.
How far from the gRNA binding site do these chemical modifications spread? It seems to me if you methylate one CpG, it's going to have a modest effect, if any. So how do you get sort of regional modification, either of histones or DNA?
Yeah, I think that's a question that's still being answered. So far, it looks like the modifications are fairly narrow, but there's a paper that I didn't discuss that describes combining Crab domains with DNMT3A fusions and DNMT3L fusions, and there they see that if you temporally order these things where you recruit Crab and then you methylate the DNA, you can induce a window of silencing that is both spreads in the same way that normal heterochromatin does and is permanent. So rather than everything I showed today, you have to keep expressing the DCAS9 fusion proteins. If you remove the expression, the methylation or acetylation marks get reset and transcription goes back on or off. This one paper is the first suggestion that we may be able to permanently control epigenetic marks, and we don't even know whether it's right or not, but if it is, it'll be if we can artificially imprint an arbitrary locus and permanently turn gene expression off or propagate gene silencing across a broad window of a chromosome, that would be really useful for modeling a lot of types of disease. But the more straightforward answer to your question is people only looked at a handful of loci and mostly using reporters, and so reporters aren't always a faithful mimetic for what's going to actually be true in the genome, and I don't think you really know what you're doing if you're just studying 4 or 5 loci. So we need some more high throughput way to measure your question. Okay, so in the last 20 minutes or so, I'm going to talk about some additional applications that people have used DCAS9 for. And so one of the things that we have been involved with at UCSF is using DCAS9 to image the epigenome. So like I said, the nucleus is a very complicated place, and people have used, Hi-C methods and other reconstruction methods to try to map 3D nuclear architecture. But this is generally done in fixed cells. So it would be really nice to be able to have a live cell way of measuring where each piece of every chromosome is relative to each other across differentiation or across different processes. And so we thought, well, why don't we try to see whether we can use DCAS9 to enrich a fluorescent signal at a region of the genome, and in this way we might be able to image a genomic locus in a live human cell. And this was a collaboration with Stanley Chi and Bo Huang. And the first thing we did was just very simply fused DCAS9 on C-terminus, or fused GFP on the C-terminus of DCAS9. We used an inducible system to keep our expression levels very, very low to reduce our background, and then we co-expressed guide RNAs off of a third lentiviral construct. So we put all this into a couple different human cell types. We tend to use cell types that are very flat. These are easier to image. So we're using mostly HeLa cells or RPE cells, which are a transformed retinal pigment epithelial cell line for these experiments. The first question we asked was whether we could image a very repetitive region of the human genome. So what we saw was that what we initially asked was, okay, we're we're trying to think of like, okay, what's the most repetitive image of the region of the genome so we can greatly enrich our fluorescence signal? And so we're thinking about centromeres, and I'm like, wait, what about telomere? The telomere, does it have a GG-PAM? And can we design a guide that'll target the human telomere? And so fortunately for us, this is the human telomere repeat sequence described here. So there are many hundreds of these repeats at the end of every chromosome. There's a GG-PAM here, and so we designed a guide that would bind to the repeats, and thereby with the expression of one sgRNA, we can target many DCAS9 GFP fusion proteins to the telomere. And so we did this, and we're hopeful we'd see local enrichment of fluorescent signal that was indicative of DCAS9 GFP binding to the telomere. And our initial experiment looked like that. It was not great. So we saw a few spots, but predominantly our DCAS9 GFP signal is enriched in the nucleolus. So you can't really see it in this picture, but in the absence in these experiments, at least, a lot of this was enriched in the nucleolus. So one of the reasons, I'm giving this talk a bit out of order, but one of the reasons that we started making changes to the guide RNA to try to increase transcription of the sgRNA or increase stability of the sgRNA was that we couldn't control transcription with a single lentiviral infection, and we couldn't image the epigenome. And in both applications, what we saw was that our sgRNA expression was the limiting factor in being able to carry out these experiments. And so if you switch to the sgRNA, the modified sgRNA design that I showed like 10 or 15 slides ago, we see that we go from very little signal at the telomere to now nice, discrete telomeric foci, and that's quantified here as the number of telomeres that you can image, or here as the percent of the whole cell GFP. So the fraction of GFP signal is actually at the telomeres. And so in all three measurements, we can see that with our improved or optimized sgRNA, we can image the telomere in a live cell. So what does this actually look like? Looks like this. The telomeres are not super dynamic. They sort of wiggle around, but we can image them. So this was a cool proof of principle. People had never really been able to. People had done this with other telomere binding proteins, but nobody had done it with a programmable DNA binding protein. And so one thing you can see here is that you get fluorescent bleaching over time, so that can be a problem for these experiments. But if you quantify the relative movement of these telomeres, you can see that although it's subtle at this resolution, they have different dynamics. So some of them are very confined within the nucleus, others seem to wander around in a random fashion, and still others have a directed three-dimensional movement that we don't understand. So it was proof of principle that this could work. We did, yep. You can take them through mitosis. Yeah, we didn't live image the entire time. We took snapshots we synchronized cells and then followed them through and imaged at specific times across mitosis. But yep, I didn't know what would happen as you get chromosome condensation during mitosis, whether DCAS9 would stay bound, because we know that it has a problem with binding to DNA wrapped around nucleosomes, but at least for the telomere and a variety of other spots, you can see DCAS9 stay bound across mitotic progressions. So telomeres are pretty specialized, and we wanted to know whether you could image additional regions of the genome. So we started by imaging some other repetitive elements of the human genome. As you can see here, this is the MUC4 locus. This gene encodes mucins that are present in your lung, and this gene, for whatever reason, has a lot of natural repetitive elements. And so we can target either an exon or an intron that have repetitive elements and show that you get 3 spots. We were really confused by this initially until we karyotyped our cells and we saw that they were triploid at this chromosome. So they were supposed to be diploid and they were not, but better to have that than have some unknown off-target binding site that we couldn't explain. So it was a good thing, not a bad thing. This, you can see, you can image 2 different regions on the same chromosome that are distant from each other. So you can see sort of 2 dim spots here. Importantly, and I don't display all the data, we did a lot of work to show that the CRISPR imaging method gives you the same information as with more standard methods like oligofish on genomic locations. Synthetic biologists are obsessed with multiplexing and orthogonal control. So we keep bringing up this idea of multiplexing and orthogonal control. We wanted to ask whether we could use different dCas9 systems from different bacterial species that bind different sgRNA structures to image two different loci at the same time within one cell. And so we fused dCas9 to a Staph aureus. We fused Staph aureus dCas9 with GFP, and then asked whether we could do the same set of experiments imaging either the telomere or these mucin genes, and we see that they're quantitatively a little bit different. The Pyogenes version works a bit better, but we can image the genome in living human cells with two different dCas9 fusions. And then if you use two fusions fused to different colors, so here we have a Pyogenes GFP fusion or a Staph aureus M-cherry fusion, you can see over here on the right that you can label a locus with two different colors. So can you guys see this? Can we turn the lights down a little bit? So that's better. So here's the take-home message for this. If you guide these two different fluorescent fusions to do two different locations along a chromosome, you can image a red and green spot. If you guide them to the same spot, you see yellow, which is as you'd expect. If you guide them to two different spots, you see red and green. So these are guided to two different spots that are 272 kb apart, and you can see that the spots are nicely separated. If you guide them to two spots that are 7 kb apart, you can see some red and green signal, but they're also partly co-localized. So this relates to the fact that these aren't single molecule imaging experiments. These are targeting repetitive elements, and but definitely we can see, what this illustrates is that you can image two different locations on one chromosome, and in theory this allows us to image in a live cell the act of chromosome looping. So you can imagine if you targeted a looped region sort of similar to here, where you have distinct spots and then induced a loop, you'd see those co-localized, and we could measure this in living cells. But we didn't get all the way there, but that's certainly something you could hope to do. Other groups have done similar things. We returned to the idea of using the RNA to recruit proteins. So another group in Nature Biotech published that you could use multiple different RNA aptamers fused to the sgRNA to recruit fluorescent proteins to image the genome in living human cells. So in this case, they fused the MS2, PP7, or BoxB aptamers to the sgRNA with 2 copies, so that each sgRNA recruits 2 fluorescent proteins, and then showed that you could image the telomere with BFP, GFP, or M-cherry. So that on its own is not that useful, but what they were able to show is that you can mix and match these RNA aptamers to create additional colors. So you can color blend with blues and greens to form the cyans, or greens and reds to form yellows, or green, red, and blue to form white. And so together this gives you 7 colors that you can image up to 7 different loci within the human genome in a living human cell. And so they actually did this. I'm still amazed that they could get all of this expressed in one cell, but with a transient transfection they seem to be able to. And so that data is shown here, where they're not imaging 7, I think they're imaging 6 colors, but they can see if you image 6 different loci, they have different dynamics. So this one sort of doesn't do anything, it stays fairly isolated. This genomic location is wandering all over the place, and we don't really know what that means, but definitely there's a lot of signal here, and it should be useful for imaging nuclear dynamics in a variety of biological applications. So another way to image, so in these imaging experiments, everything has been done with defined guides, where we express one or a set of guides that are cloned in low throughput. Rebecca Heald's lab here at Berkeley wondered whether you could image a whole chromosomal region. And so they derived this sort of clever scheme for building a large number of sgRNA expression constructs from a desired region of the genome. It's described in this paper, but basically you can use PCR to amplify regions of the genome, and then you digest with this specialized restriction scheme such that you can ligate all of the digested DNA into an sgRNA format and express thousands of sgRNAs that tile along a chromosomal region. And so here they're doing this in Xenopus frog eggs, and you can see that here's the nucleus, and then they're expressing, I forget the exact number, it's I think on the order of 1,000 sgRNAs targeting this region of the chromosome, and you can see a very, very bright spot where that chromosomal region is. So I showed you one of the things that we struggle with in imaging experiments is always fluorescent bleaching. So when I showed you the telomere movie, you could see over time it bleaches. So the more signal you have, the more experiments you can do, or the more exposures you can take. And so this is a way of enriching signal, and is also a very, very cheap way to image a whole chromosomal location. So along the lines of worrying about bleaching, we wondered whether can we, yeah. So like if you did a pull down for that locus and then used mass spec to measure protein occupancy? For the telomere, we mostly know what's there. Those are very well characterized. So in that sense, yes. For an arbitrary locus, it's been difficult to use dCas9 to pull down a locus and measure the proteins present, because for every you have 2 on-target binding sites, but we're frequently expressing a lot more than 2 dCas9 proteins, and they're searching across the genome at putative low affinity interaction sites. So anywhere where people have looked by ChIP-seq, anywhere where you have a PAM plus 7 base pairs of homology to the sgRNA, you can get enrichable signal by ChIP-seq. And so that results in thousands of binding sites that are measurable across the genome, and the aggregate signal from the off-target binding sites that are very transient is quantitatively more than the 2 on-target binding sites. And so it's hard to see the signal from noise. There may be other ways to do that, but people have struggled to do that. Okay, so we wondered whether we could get more sensitive imaging or single molecule imaging by recruiting multiple fluorescent proteins with one sgRNA. So this is a crude diagram of the SUNTAG, the scheme I showed you before with our second generation CRISPR-A system, where we can fuse to any protein a series of epitopes. If we co-express a single chain variable fragment fused to another protein, we can enrich either an activation domain or a fluorescent protein out of arbitrary sequence. This works because you can express a lot of repeats, and the binding is very, very tight for the epitope. And if you do FRAP experiments, the binding is very stable. So these systems were being used by Ron Vale's lab to image kinesins. So this is a movie of kinesins moving in a living human cell, where you fuse these epitopes to a kinesin, and then you co-express the SFE-GFP fusion, and you recruit up to 24 GFPs to an individual kinesin. And so if you motion blur out the background, you can image single molecule image individual kinesins moving within a cell. And so this is an illustration of how we can use a multiplexed enrichment for fluorescent signal to increase our sensitivity and do single molecule imaging. We haven't actually done exactly this with dCas9 for a couple of technical reasons, but the potential is there for doing single molecule imaging of an arbitrary locus of the human genome, even if you don't have a repetitive sequence. So you can, these are just nice movies, so I included this, but basically this is a blown up this is a blown-up view of this, and the arrows are tracking single kinesin imaging, moving along a microtubule. So pretty cool. I was sort of new to the field of single molecule imaging, but there's a lot of interesting cell biology you can do. This can be used with dCas9 as well to greatly enrich your sensitivity. So this is our version 1 CRISPR imaging system where we're imaging telomeres. You see spots. This is version 2 where we're with each sgRNA recruiting 24 GFP molecules, and you can see that the signal is basically blown out. So we have way more sensitivity if we recruit many fluorescent proteins with a single sgRNA. So moving out of the nucleus, you might wonder, well, what else could we use these programmable binding proteins for? And could we use this to track RNAs as well as and not just DNAs? And this is really a special interest of Jennifer's, actually. And so there are reviews published about how RNA localization impacts a lot of different kinds of biology. And people for the last 15 or 20 years have been using these RNA aptamers like MS2 that I've talked about multiple times today to recruit fluorescent proteins to RNAs to either image them or to pull them down and study what's associated with them. And so these are summarized here, but we wanted to know, well, could we use a CRISPR system in a similar way to bind to an unmodified RNA? So all these RNAs have to be modified where you append multiple artificial binding sequences. And so there's always this issue of, well, you changed the RNA, is this actually really what's happening, or do you have some artifact that's associated with this chimeric RNAs? And so Jennifer's lab around 2014 started to explore whether you could use Cas9 to bind to RNA molecules. And what they were able to show is that if you trick Cas9 by providing what they call a PAMmer in trans, so that Cas9 normally binds double-stranded DNA, right? It's not supposed to bind single-strand DNA or single-strand RNA, but if you provide a PAMmer in trans, and that's detailed in this fairly complicated experiment, you can get Cas9 to bind to a single-stranded RNA and cut it. So that's shown down here where if you apply a PAMmer in trans, it'll bind and cut. This is a similar experiment using dCas9 showing that you get binding of dCas9 to single-stranded RNA only in the presence of the PAMmer. So here's binding to double-stranded DNA. You can see it's very robust. Here's binding to single-stranded RNA. It's very, very weak. If you add the PAMmer to trick Cas9 into thinking it's binding double-strand DNA by forming a duplex between the PAMmer and the single-strand RNA, now Cas9 will bind and cut. And so this was used early on for IPs, so you can immunoaffinity purify cellular RNAs using a Cas9 plus a PAM. And this works well for high abundance RNAs, and is, I think, in my understanding, is still a work in progress for very low abundance RNAs, and intermediates are sort of in between. But it's a new tool for pulling down endogenous cellular RNAs to study what the set of proteins that are associated with them, or a variety of other things. We want to do more than just IP RNAs, probably, and so in a collaboration with Jennifer, Jean Yeo, I don't know how to say his name, lab at UCSD showed that you could use a similar Cas9 scheme where you're supplying both Cas9 or dCas9 plus a PAMmer to image cytoplasmic mRNAs. So the data is shown here. Essentially what they show is that if you express a dCas9 GFP fusion with a nuclear localization, you see that most of it goes to the nucleus. If you express an sgRNA with the dCas9 GFP plus a PAMmer that targets this GAP-DH mRNA, you can see that you get relocalization of this dCas9 GFP fusion out of the nucleus into the cytosol. So here you have very little GFP signal in the nucleus, and most of it's cytosolic. And they did a lot of controls to show that the GFP signal in the cytosol is predominantly bound to the target GAP-DH mRNA. And so you can do a little bit more sensitive microscopy to track actin localization, and that's shown here where you can see actin localization in the cytosol. I think this remains a bit of a work in progress in that for these technologies, you can definitely pull down and visualize abundant RNAs. Very low abundance RNAs remain a challenge, but certainly promises to be an additional kind of application for what these could be used for. Personally, I think that Cas9 will probably not be the winner for imaging or pulling down RNAs. I tend to think that there's going to be a different CRISPR